To understand how the world we see around us today has come to be, we need to go back to the very first dynamics of the universe. In the first 3 minutes after the Big Bang, the elements hydrogen, helium and lithium were formed. Here's an illustration of the formation of helium from hydrogen atoms or free-floating protons. This process is called hydrogen burning. After these first minutes, nuclear reactions such as this one became no longer possible. This was the case because the universe cooled down as a result of its rapid expansion. In our current universe, however, there are many other, heavier elements besides hydrogen and helium, which are crucial for life. So where did all those elements come from? The answer literally lies in the stars. As gravity started to clump together, big balls of hydrogen and helium stuck together and stars were born. The stars became very hot due to all the gases pressing together. At this point, nuclear reactions happen again. Most of its life, a star will fuse hydrogen into helium. This fusion creates radiation that keeps the star from collapsing. When the star runs out of hydrogen, the gravity wins and the core of the star will shrink, while the outside layer will blow up. The core of the star will now become even hotter, and the possibility to form heavier elements now arises. All the elements up to iron are formed to fuel the star. The star is now like an onion, the inner layer iron and the most outer layer helium. At the end of its life, the star will explode. That explosion releases so much energy that almost all other elements are made, except for the artificially synthesized ones. We now know where the special building blocks of all matter came from. So from the elements made by stars, how did life emerge? Out of two theories, the first one is the RNA world hypothesis. A special RNA molecule called the ribozyme can self-replicate and catalyze certain reactions. Eventually, when the RNA fragments got enclosed by a membrane, these protocells were able to maintain a different inner environment and they divided by binary fission. The other approach is the metabolism first theory. This states that life began around the hydrothermal vents in the ocean. In these hot, metal-rich conditions, there arose metabolic pathways, catalyzed by metals. Amino acids and peptides also formed. Once the pathways get enclosed by membranes, they become protocells. These protocells need to have acquired genetic information to undergo natural selection, but this is still unclear. Eukaryotic cells are far more complex than the cells of archaea and bacteria, and their origin can be explained by the endosymbiotic theory, which states that prokaryotes engulfed each other multiple times. Another component of the origin is the viral eukaryogenesis theory, where an archaeon takes up a bacteria and gets infected by a large DNA virus. It took another 1.5 billion years for eukaryotes to become as diversified as they did in the Cambrian explosion. Say that you have a mouse, a typical multicellular organism. You zoom in on its back to its epidermis and see layers of cells, all containing identical copies of DNA in the nuclei. How then, considering that these cells came from one original egg cell, do these cells know what to be? How would an embryonic cell know to develop into a nerve cell, for example, or a red blood cell? The answer doesn't lie directly in the genome, but above it, in the epigenome. The epigenome consists of all chemical compounds added to the entirety of one's DNA that influence gene expression throughout the lifespan. And this can occur as a methyl group attaching on DNA or histone packaging of the DNA strand. In particular, gene segments packed tightly together by histones cannot be transcribed and expressed. When the packaged DNA is loosened, chemical tags can attach to the histone tails to further influence gene expression and repression, making a sort of histone code. Methylation on a nucleotide-based cytosine, for example, knocks out a hydrogen atom and inserts a methyl group. A nucleosome is made of DNA wrapped around an 8-histone core, and individual histones are labeled based on the compounds usually attached to their tails. Epigenetic influences can be inherited by offspring because although reprogramming cycles erase most tagging, some remain. That means that any environmental or behavioral factors that epigenetically affect an organism's DNA might carry on over several generations.